Hello, everyone. This is Patrick Morrison from the Wolfpack Astrobiology podcast, and in today's video series, we'll look at the stem of life, covering how we got from the first organisms to the last common ancestor of everything alive today. We have all seen images meant to depict Darwin's tree of life, where everything alive today can trace its origin to a common ancestor. On the metaphorical tree, this is where the branches start to grow away from the trunk. However, what's often lost is that this wasn't where it all began. If we were to adhere to the metaphor, life began at the base of the tree, while the roots represent prebiotic chemistry. The trunk is everything that happened between the origin of life and the last universal common ancestor, also known as LUCA. An approach often used by astrobiologists is to combine the top-down and bottom-up approach to study the origin of life. The bottom-up approach studies prebiotic chemistry, while the top-down approach utilizes comparative molecular biology. The problem with combining these approaches is that they lead you to very different places. This isn't because either is inherently flawed. It's that the top-down approach leads you to LUCA, while the bottom-up approach will take you to the origin of life. And between 200 and 500 million years passed from the first cells to the common ancestor of everything alive today. To sum up what happened in that time period, life went from a simple set of self-replicating reactions to something on par with modern prokaryotes. Needless to say, that is a lot. Modern life is divided into two superdomains, bacteria and neomora, which includes archaea and the eukaryotes. The origin of life domain, or OLD, refers to a functional domain that includes everything that came after the origin of life and before LUCA, and this can be thought of as the trunk or stem of life on Earth. So, how do we infer what happened in the time of the origin of life domain? Often, phylogenies of individual genes are constructed and compared with the phylogeny of the organisms which they are obtained from. If the organisms were from all extant branches of the tree of life, and the shapes of the phylogenies are similar, the gene likely existed in LUCA. This approach on its own can only be used to identify which genes LUCA had, but if you identify a series of genes in a gene family, all of which were present in LUCA, you can construct a phylogeny of those genes to determine in what order each evolved in OLD. Although an understanding of early life should not be based around a bottom-up approach, it is useful when metabolic pathways resemble abiotic processes. Because OLD existed so long ago, including a time where metabolic reactions weren't catalyzed by enzymes, there are often times where no homology exists between enzymes which catalyze specific pathways. However, similarities in the pathways themselves and reaction mechanisms can help to infer how the evolution of various pathways is interconnected. One novel, but still controversial technique used to study the history of life which takes lateral gene transfer into account is the rings of life. This technique has been used to study the origin of life domain by looking at the gene families which were present in LUCA and turning the clock backwards to determine in what order various systems came into picture within the first organisms. Going back to the start, what were the first organisms like? To recap the Origin of Life series, the first cells would have arisen sometime between 4.3 billion years ago, when liquid water first existed on the surface of Earth, and 3.8 billion years ago, when the oldest biosignatures, carbon-13 depleted graphites, were identified. Life has three requirements: replication, compartmentalization, and metabolism. In the first cells, compartmentalization would be provided by bilayer fatty acid membranes. Replication would be allowed by RNA oligomers within the cell, which could be duplicated, and metabolism would be facilitated by catalytic mineral surfaces. The cradle of life on Earth was likely terrestrial serpentinizing pools, where the cellular biology of the first organisms was largely dependent on wet, dry, hot, cold, and acidic basic cycles. In the Origin of Life series, we gave an example of what a minimal first cell was like. Here, we'll start with a maximal protocell, which has a combination of ligase, phosphorylating, and polymerizing ribozymes, a membrane composed mostly of carboxylic acids, but also some phospholipids and aminolipids, and a series of iron-sulfur cluster nanocrystals with varying impurities of either nickel or cobalt. The features of the membrane and catalysts would, at the onset of life, be determined by the local geochemistry, and, apart from the reactions catalyzed by the ribozymes, the metabolic reactions would be carried out via abiotic mechanisms. One variety of prebiotic or early biotic catalysts that are often overlooked are vitamins. In many modern enzymes, vitamins act as cofactors, which enhance catalytic activity. When ribozymes are evolved in laboratory settings, vitamins perform very similar functions. B vitamins, in particular, are tied to life's core metabolic activity, and most B vitamins can be synthesized abiotically under early Earth conditions. The exceptions are vitamin B1 and B7, but an important caveat to note is that over the past few decades, we've discovered mechanisms by which other B vitamins could be synthesized abiotically. So, we may yet discover a way those two could be synthesized. Furthermore, B vitamins often catalyze the production of other B vitamins, and this may have established an autocatalytic network in the first organisms. Autocatalysis refers to when the product of a reaction itself acts as a catalyst, causing the rate of the reaction to accelerate as long as reactants are provided. So, once a protocell obtained all the B vitamins, it would not only facilitate the production of biomolecules such as lipids, amino acids, and nucleotides, but establish a self-sustained and expanding network. This may have acted as a key intermediate between the first cells, which would have been tied to the local geochemistry, and autonomous organisms. In terms of how individual B vitamins aided in early metabolism, B2, flavins, B3, niacin, and one found in prokaryotes known as F420 facilitates redox chemistry. Vitamin B1, thiamine, aids in carboxylation reactions, while vitamin B12, cobalamin, does the same for methyl transfers. An interesting feature of vitamin B12 is that it is based around a siroheme group. Sirohemes are used in many metabolic processes today, such as photosynthesis and respiration, and the progenitor of these was likely B12. Roughly 20 years ago, many thought that ribozymes acted as the biocatalysts for the first organisms. However, limitations in the number of reactions that they could catalyze has called this into question. Some metabolic reactions that could have been carried out exclusively by ribozymes include the ligation and polymerization of RNA sequences, along with the phosphorylation of small organic molecules to be fed into early metabolic pathways. But instead of an RNA world, biochemists have put forward the notion of a ribonucleopeptide, or RNP world, as a precursor to modern biochemistry, which is catalyzed almost exclusively by protein enzymes. The addition of single amino acids to ribozymes can increase both their stability and catalytic repertoire. A ribozyme would have catalyzed the first step of a given reaction pathway, and as it diversified into a ribozyme family, which catalyzed each step of the pathway, the amount of amino acid cofactors, or the degree of peptidization, would have increased. Apart from acting as cofactors, amino acid monomers can act as catalysts themselves. For one, L-amino acids catalyze the production of D-glyceraldehyde, a precursor to sugars, from glycoaldehyde and formaldehyde. Modern organisms use the L-isomer of amino acids and the D-isomer of carbohydrates, and that can partially explain why this stereochemistry is used. Glycine and diglycine catalyze the production of dipeptides, and since diglycine itself is a dipeptide, there's potential for some more autocatalysis. Wet-dry cycles can produce di and tripeptides, which would have exponentially increased the possible catalytic and structural functions available to early life. While biocatalysts would have increased both the rate and efficiency of metabolic reactions, a lot of it was probably driven by inorganic metal-based mineral catalysts. A perspective on the origin of life which includes these could be called the RMP world, where M stands for mineral. Today, many enzymes possess metal cofactors which enhance their reactivity. A particularly common variety are ferredoxins, or iron-sulfur clusters, which facilitate redox reactions. These clusters have a similar crystal structure to iron-sulfur minerals, most notably gregite and mechanowite. If an amino acid was phosphorylated, it can bind to a nanocrystal of one of these minerals, which would keep it suspended in solution and possibly aid in catalysis. Divalent manganese, zinc, and magnesium ions often act as cofactors in ribozymes, modifying their activity. Furthermore, magnesium, along with iron, can facilitate the transfer of organophosphate groups from one molecule to another. Magnesium cofactors are often used for this purpose in modern enzymes, while iron is not. In many reactions involving organic chemistry, there is a major product resulting from the reaction, which comprises most of the individual molecules produced, but there are minor products as well. These minor products will be produced in abiotic analogs of various metabolic pathways and can't be fed into the next step of the pathway. In the pre-TCA cycle, the presence of zinc and chromium ions will decrease the production of minor products and thus increase the pathway's efficiency. Copper, nickel, cobalt, and vanadium ions catalyze Strecker reactions, which bind ammonia to alpha keto acids to produce amino acids. And finally, boric acid will lead to enhanced amino acid polymerization during wet-dry cycles.